A-level Biology 9700, Paper 4, Variant 2, October, November 2025. It's question number 10, the last question of this paper. So we will discuss it right now. The question is, nerve impulses can be transmitted along a myelinated motor neuron to a neuromuscular junction at fast speed of up to 100 meter per second. Outline how a transmission speed of 100 meter per second is achieved by the neuron. So we have to give the outlines for this. That is the how it happens. So that I would like to say in when there is saltatory conduction in myelinated neurons, the axon is insulated by a myelin sheath, you know very well, which is formed by the Schwann cells. So this sheath is impermeable to ions, so due to which depolarization is cannot take place in the myelinated sections. So after the depolarization and action potential can only occur at the gaps in the myelin sheath known as the nodes of Ranvier, where voltage gated ion channels are concentrated. So in saltatory conduction, the local circuits jump from one node to the next. So this is much faster than the continuous wave of depolarization seen in unmyelinated diameter. So while not explicitly mentioned in the prompt, a larger axon diameter also reduces resistance to local current flow. So it contributes to the high speed. So this is the whole, you can say, answer of this as it is of 3 marks. So the, the points which I am going to write, first of all, we know that the axon is insulated by myelin sheath, which is formed by the Schwann cells. So this sheath is impermeable to ions, preventing depolarization. So depolarization and action potential occur only at the gaps in the myelin sheath known as nodes of Ranvier. The local circuits jumps from one gap or node to the next fast. One thing is also mentioned that a large axon diameter also reduces resistance to local current. Current flow contributing to high speeds. Now, part second of this question is, when a nerve impulse reaches the neuromuscular junction, acetylcholine is released and diffuses to the sarcolemma. Describe how the release of acetylcholine can result in the binding of calcium ions to troponin in the sarcomere. So the part B is here, as I would like to explain it, that what happens. First of all, the binding and the depolarization takes place as acetylcholine diffuses across the synaptic cleft and binds to receptor on the sarcolemma, which is on postsynaptic membrane. So this opens ligand gated sodium channels and causing an influx of sodium and depolarizing the membrane. So action potential speed becomes propagates along the sarcolemma and deepens into muscle fiber. So T tubules are and also via T tubules, then sarcoplasmic reticulum. Uh, what happens with them? That is, the action potential in the T tubules triggers the opening of voltage gated calcium channels in the membrane of the sarcoplasmic reticulum. So the calcium ions release. Calcium ions diffuse out of the sarcolemma into the sarcoplasm, and then the increased concentration of calcium into the sarcoplasm allow the ions to bind to subtroponin. So I would like to say that this binding causes a conformational change in troponin, which pulls tropomyosin away from the myosin. Binding sites on the actin allowing attraction concentration to begin. So contraction takes place. Now I would like to write its answer in detail because they have asked description. Question is of 4 marks. So acetylcholine diffuses across the synaptic cleft and binds to receptors. Binds to receptor on the sarcolemma. This opens ligand gated sodium channels. Enzyme the influx of sodium which depolarizing the membrane. Which generates this depolarization generates an action potential that propagates along the sarcolemma. The sarcolemma and deep into the muscle fiber via the tubules. The action potential in the T tubules triggers the opening of voltage gated calcium channels in the membrane of sarcoplasmic reticulum. Calcium ions diffuse out of sarcoplasmic reticulum into the sarcoplasm. Now, ions bind to troponin molecules located on the actin filament. Now the last part of this question, part C. Muscle contractions can be affected by low blood glucose concentration. Suggest how a little blood glucose concentration would affect the functioning of the sarcomere. So here muscle contraction Uh, First of all, we have to give suggestion about this. That is, the question is about the uh, metabolic supply to the mechanical action of the sliding filament theory. You know that breakdown. Um then you can see that the glucose on the impact of low glucose blood glucose on the sarcomere. So I would like to write this question as low blood glucose means that less glucose is available for glycolysis and subsequent stages of the aerobic respiration. So this results in a decreased rate of ATP synthesis in the muscle fibers. So, due to this, ATP is specifically required to break the cross bridge between the myosin head and the actin filament. Without sufficient ATP, myosin heads remain bound to actin, preventing the resetting of the sarcomere for the next power stroke. So, ATP hydrolysis provides the energy for the myosin head to cook back into its high energy state. A lack of energy means the myosin heads cannot effectively pull the actin filaments toward the M line, leading to weaker or failed contractions. So, ATP is also needed for the active transport of calcium ions back into the sarcoplasmic reticulum. If calcium remains in the sarcoplasmic, because the bumps lack energy, the binding sites actions stay exposed, which can lead to muscle fatigue or comparing rather than controlled contraction. So, briefly, you can write all this because the question is of 3 max. Low blood glucose leads to a decreased rate of respiration, resulting in less ATP being produced in the muscle fiber. Less ATP is available for the detachment of myosin heads from actin binding sites, preventing the cross bridge cycle from continuing. Meaning actin filaments are not pulled not pulled effectively, resulting in a failure of the sarcomere to shorten. So it was all about this question and this paper as well. Please like, share, and subscribe my channel. Please also share with the other students, your junior students, your senior students, uh, to visit my channel. They in this way they will be able to find more videos on more papers. Thanks for watching.